Australian researchers have uncovered a new way that bacteria could become superbugs and avoid being killed by antibiotics. Joining us live is Dr. Tim Barnett, lead author of the research and laboratory head at the Telethon Kids Institute. Appreciate your time, thank you. Let's just start at the beginning here. When we talk about superbugs, what are we actually referring to? What are superbugs and why do we need to be worried about them?
Yes, thank you. So superbugs are bacteria that cause infections, and they have evolved resistance to antibiotics, which are the main way that we treat these infections. Generally, when we refer to superbugs, we're talking about these pathogenic bacteria that have evolved resistance to multiple antibiotics, including the first-line treatments for these infections.
So what does this latest research tell us about how these superbugs seem to be getting better, it sounds like, at avoiding antibiotics?
Yeah, so what we've found is a new mechanism of antibiotic resistance, but what is different about this is that it's not easily detected in a pathology lab. It's a resistance that is only really functional during an infection. And so when a pathology lab tries to look for resistance, the bacteria will test sensitive, but then when they're causing an infection, they'll be resistant.
Okay, and did the research come up with any ways to get around that?
Yeah, well now that we've uncovered the mechanism for this resistance, this type of antibiotic resistance, we've defined the gene that causes this resistance mechanism. So we can now develop tests to identify the gene itself. And we've also identified a way that a compound we can add into the pathology lab test that will make the bacteria resistant. So we can detect it both through their ability to grow in the presence of the antibiotic and also using the gene that we've defined.
So it sounds like amazing work. More broadly, are doctors over-prescribing antibiotics, do you think, in Australia? Is that one of the problems here?
Well, that's a very big and complicated question. There's definitely over-prescription of antibiotics in certain circumstances, and really, when as patients we need to not always ask for antibiotics whenever we've got a runny nose. We have to save the antibiotics for when we really need them. We know there's a lot of antibiotic use that's for example in agriculture and things like that can contribute to antimicrobial resistance. But it's a very big problem, and we really need to kind of understand the best ways to minimise antibiotic use. One of the ways we can do that is through better identifying whether an infection is resistant or sensitive, so that we can reserve antibiotics, so we can treat with only the best antibiotics that will benefit that infection.
Yeah, so you mentioned often it's the patients pushing the GP essentially to give them the script for antibiotics. Should parents in particular be thinking twice about giving kids antibiotics if their doctor doesn't seem convinced that they'll work? I think we've all probably had that situation where the GP might not be 100% if the antibiotics would be the answer to a child's particular illness.
Yeah, it's a tough question for me to answer as a scientist who studies mechanisms of resistance and things like that. I think parents obviously have to respect the doctor's advice, because there are definitely infections where antibiotics are important to prescribe. But whether a particular infection requires antibiotics or not, that's the decision of the doctor.
So in terms of this resistance being a sort of global public health risk, what are the key threats globally on this front?
So there's a number of threats. Really, we need to understand, we need to track antimicrobial resistance pathogens as they cause infections in the community and disseminate around the world. That can be through either just monitoring infections as they come through pathology labs and seeing whether the infections are caused by resistant or sensitive bacteria. We can also do this through techniques that are more commonly being used now, such as genome sequencing to detect the specific genes themselves. But what I think our work really highlights is we also need to do discovery research to understand the mechanisms and identify new antibiotic resistance genes that can then be fed into these other ways to detect resistance. I suspect that our discovery is really the tip of the iceberg. There's going to be other mechanisms of resistance where the bacteria are only resistant during an infection, and we need to do research to understand this.
Dr. Tim Barnett, really appreciate you taking the time to explain all of that for us. Thank you very much.
Thank you so much for having me.